have to discuss some questions as well. If you want to spare some time, you have to pay attention so that I can finish it. Okay? Okay, so I have brought some questions for you to discuss and have an idea what sort of questions could be. So, it stops DNA replication because it becomes a pseudonucleotide. Okay? And this enzyme, IMP dehydrogenase, which is involved in generation of thioguanosine from thioinosine. This enzyme could be blocked by mycophenolate. So, in a nutshell, if you give azathioprine, you end up having pseudonucleotides, okay, thioguanosine. And if you give mycophenolate, you effectively block the conversion of natural inosine to guanosine. Natural. So you don't give them together. No. This IMP dehydrogenase is the natural enzyme. As I said, the drug uses the natural enzymes present already. Ammar, what's wrong with you?
Explain.
Please come and sit over there, or here somewhere, please. Any further cross-linking, you will be expelled out of the system. I mean, out of the class. Okay? So, the mercaptopurine, like any other purine, undergoes metabolism and is broken down into by xanthine oxidase to 6-thiouric acid. Yes?
So you mentioned we don't use these
together. No. Why you use them together? Because why you don't use them together, there's no use. I mean, utilizing the same pathway over and over again. If you want to have additional drug, it should have some different mode of action.
So they have the same.
So that they synergize rather than the additive. Okay? So azathioprine, let's see. Yes, you know. You know this now. So, now this is something to discuss. Diarrhea is the major adverse effect. And whenever you use it with allopurinol, last time we discussed it also, and this time also, that since it's a purine, it will be removed. The azathioprine being a purine will be metabolized into 6-mercaptopurine, which will then be degraded and removed from the system by xanthine oxidase. So if you block xanthine oxidase with allopurinol, which is a drug used in gout, what will happen? The drug would remain in the system for the prolonged period of time. So there will be bone marrow suppression, there will be, exaggerated adverse effects. So you reduce the dose of azathioprine to 1/3 or 1/4. Okay? 1/3 or 1/4. You don't reduce allopurinol, you reduce azathioprine. Now mycophenolate, it's a salt. Mofetil is removed, becomes mycophenolic acid, which is the active moiety. Inhibits inosine monophosphate dehydrogenase and hence deprives the synthesis of guanosine monophosphate. You already know it. Toxicity is diarrhea, the main. It is teratogenic, but less so than azathioprine. Because of it's an acid, and can be given oral, so don't use it with antacids. Okay, they would be neutralized and will be removed from the system. They won't be absorbed. Now here is, please. Now, something to understand. Immunoglobulins. I know that you already have been through the structure of immunoglobulin. Immunoglobulin being immuno means from the immune related. Globulin because it's a protein. Globulin it's a globulin protein. Okay, and it has got heavy chain and light chain. Okay. Now, heavy chain is the one which forms the fixed region, which fixes to the cell, for example, mast cell, FC region. Whereas the light chain, along with the variable region of the heavy chain, it forms the antigen recognition portion of the immunoglobulin. This is called FAB. Okay, antigen recognition end of the immunoglobulin, FAB portion. Now, immunoglobulin is a protein, you know, that means it has got its own gene. So immunoglobulin gene, okay? Now, immunoglobulin gene is the only example where mutation constantly takes place and it is not corrected.
It has to be.
It shouldn't be.
Yeah, it has to be mutated.
It has to be mutated, okay? Because we need to have lots many types of immunoglobulins, okay? So an IgG against one antigen would differ from the IgG against another antigen, for example. So this is a very good example of a continuous site of mutation which is purposeful, beneficial for the organism. And it is not corrected. Okay? Subhanallah ta'ala. And this immunoglobulin gene, that's why is called as V gene or variable gene. Okay? So the proteins which it codes for, they are termed as V region or variable region. Okay? So the hands of these two hands of the FAB portion, yeah, they contain some portions of the proteins which are continuously variable. Okay? So they are called as the CDR regions of the variable region. CDR of the variable region. Clone determinant region of the variable region. Okay? So CDR region for the variable region. So the actual recognizing region is CDR in the variable region. So the variable region has got the CDR, means which varies actually, and a non-variating sequences of protein as well. What is important? We'll see next slide. We'll see the next slide. Now here is 4 types of immunoglobulins. Depending on their types of monoclonal antibodies, which are synthesized in the labs and used clinically. Now the completely orange one is the one which is murine, mouse origin. Murine, I mean just like bovine. Bovine is what? Cow origin. So for mouse origin is murine. Okay? So completely murine, for example, muromanab. Okay? All mouse. Then technology advanced. The next group of antibodies is where we have the fixed region, FC region is human, okay, and coupled to the murine variable region. And this is called chimeric. Okay, what is chimera?
It's an animal, mythical creature.
Okay.
Different body parts.
I don't believe in the zodiac signs, okay, but some of you would be interested in zodiac signs. One of the zodiac signs is Sagittarius, for example. Okay? That has a horse body and a male torso attached to the inside of the neck. It's a chimera. Okay? Human and horse chimera. It's a hypothetical animal. Okay? So chimeric means a mixture of 2 different species. So this is half murine, half human. Okay, chimeric. The example is infliximab. The variable region is murine. Fixed region is human. Technology advanced further, and now we have only within the variable region, only the CDR is murine. All other parts of the immunoglobulin is human. So now it resembles more human. So this is humanized. Not human. Humanized. Example, rituximab. Okay? And the final is complete human. Okay? And the example is adalimumab. Okay? This is human, not humanized. Human. Okay? So, but they are not produced by humans. They are produced in labs by microbes, which are genetically engineered, where the genes from, similar to humans, they have been transfected. And then they starting producing the human proteins. Just like insulin. Remember the insulin?
Yes.
So recombinant DNA technology. Now let's see. Now this is the chart which gives you an idea at what target we have drugs.
So this is summary?
Yes, it's summary. So daclizumab, so you have drugs against interleukin-2 receptor. Okay, this is an antibody. You have an antibody against the TCR complex. Okay, T cell receptor complex. They have drugs against the calcineurin, mTOR, inosine monophosphate dehydrogenase, DNA, nucleotides rather, that would be more specific, and finally the GRE in the active DNA. And here is the action. These are the references as well as the disclaimer. Now let's pay attention to clapping at the end, please. So let's finish the remaining part of the anti-cancer. We couldn't complete immunoglobulins, anti-cancer immunoglobulins. Okay? Now, immunoglobulins and tyrosine kinase inhibitors, you can see trastuzumab. Now, these are the ones which you have. These are the anti-cancer immunoglobulins. Trastuzumab, now you see this is humanized. Now you know what is humanized?
Yes.
Okay. Cetuximab is a climatic. Now you know what is climatic?
Yes. Yes.
Okay. Bevacizumab is humanized. Okay. And imatinib is just monoclonal antibody or mouse. And what is it doing? Let's see. Imatinib blocks the tyrosine kinase. Okay, which is the platelet-derived growth factor receptor coupled. Okay, HER2 is the epithelial-derived epidermal growth factor, and it blocks trastuzumab blocks this is the monoclonal antibody which blocks the receptor. Okay, of the HER2. What is the most breast cancer? So breast is the skin appendage, and skin appendage epidermal. Yeah, you know. Yeah, the breast is a skin appendage. So it responds to epidermal growth factor. So EGFR is the epithelium-derived growth factor. Cetuximab takes care of by blocking the receptor. And the VEGF, vascular endothelium growth factor. Here, the bevacizumab binds the factor, not the receptor. So this is the difference. So it inhibits the angiogenesis. So it's an anti-angiogenic, ultimately. Bevacizumab. So now let's try to discuss some MCQs, if you are interested.
Yes.
Are you?
Yes. If they come to exams.
Of course they won't.
You will upload
So I won't read them on this, neither I will be uploading them. But
we will read them on study. You will upload them?
But here we have, just as an understanding, some of your colleagues yesterday or day before yesterday, they requested for an idea how they should be approaching the MCQs. Okay? So I discussed it with the section head, Dr. Dilip, and he kindly granted permission to present some MCQs as an example. Okay?